Welcome to another Anatomy and Physiology II Bio 265 video in a nutshell.
Today we are going to be talking about the cells of a specific immunity, how they're activated, and once activated, how they function. This represents a macrophage. A macrophage is part of the non-specific immunity, and it will locate anything that's bad. In this case, let's say that it's a bacterium. So it will locate the bacterium, it will eat the bacterium, and then after breaking the bacterium apart, then it is going to take a piece of that bacterium and it's going to express that piece on a receptor on its surface. MHC2, if anyone's curious as to what it's called. So it's going to pick a piece of bacterium and it's going to put it on its surface, and that will be the antigen, or the recognizable piece of the bacterium. So this little bacterium piece is going to be recognized by a helper T cell. And you can think about the macrophage kind of like the general in the army. He's the general, he picks the target, he picks the bacterium, he picks the piece of the target that he wants everyone to go after, and then the T helper is kind of like the messenger on the horse. It comes over, it takes a look at the message from the macrophage, it takes a look at the antigen, the piece of bacteria, and it says, oh, I got the message, off I go. So the T helper messenger comes over and it will come to either a B cell or to a cytotoxic T cell, and it's going to show them the message. It's going to show them basically what their target is. It's going to activate them, so they are now off to go look for whatever the bad guy is. So when these two cells get activated, they fight bad guys in different ways. We're going to focus first on B cells and how they're effective in fighting a bad guy. B cells, their method is they produce antibodies. Antibodies, we typically see them drawn kind of like this. They look kind of like a Y. They actually come in many different shapes. But what's important to know is that these antibodies are sticky or have little receptors on their ends. And they are sticky for this particular antigen. In other words, if this is our B cell, it's going to produce these little antibodies and it's going to spit them out. And every one of these antibodies is specific or will bind onto this particular antigen. So in other words, if let's say here's the antigen on this bacterium, if there happened to be an antibody floating by, here it comes, it's going to recognize its matching puzzle piece, or it's going to recognize what it's supposed to stick onto, and it's going to hook onto that part of the bacteria. So these antibodies, once they bind to their target, once they find their matching puzzle piece and stick on, alone they're just a piece of protein, but they help other things to happen. First thing that they're able to help happen is enhanced phagocytosis, or in other words, it's kind of like a big waving red flag to all the other macrophages and all the other phagocytes in the area that says, this guy's bad, come get him now. The second thing that they are able to do is they are able to activate complement protein. And complement protein, just like macrophages, is part of the non-specific immunity. So again, here we have a crossover. But these antibodies are going to activate complement protein. And that complement protein, it's going to come and it will poke holes in the bad guy. So again, it's a big waving red flag. Antibodies on here, it says, come poke holes in this guy. And the complement says, I see my target, here I come, and start stabbing holes in the bacteria. And the third thing that it can do is it can neutralize the target. So let's draw another bacterium, and let's do this one in a different color. So here's our new bacterium, and let's say that this bacteria has a little receptor guy here it wants to use to get into a cell and infect it, and then it has another important little receptor here that it wants to use as a docking station, and so it's floating through and it's looking for cells that can bind to, or things that can hook onto and infect them. So let's say that a macrophage eats it, and we go through the whole process, and we now have antibodies that bind to this bacterium. So here comes our little antibody floating along, and says, oh, there's my target, sticks on. And then let's say that there's another piece that it sticks onto over here, and then there's an antibody that hooks onto this one. But eventually what happens is pretty soon this bacterium gets surrounded by antibodies that are hooking onto it. And that's effective because these receptors can no longer be used if they're bound to directly. Or let's say that the receptor happened to be right here on the side, and it wanted to, so here's the receptor, and let's say that it wants to hook onto this cell right here. But every time they try and get close to each other, it can't, because there are antibodies in the way. kind of holding it at a distance, if you will. So if you have all these antibodies, every time things try and bind, they bump into the antibodies. It's kind of like surrounding it like a pincushion. Nothing can get close enough to bind. So basically this bacterium is now neutralized. It can't bind to anything, it can't function as it should, and it just floats around in the bloodstream until finally a macrophage gets a hold of it and eats it up. So that's how a B cell is effective, with these antibodies that do those three functions. A T cell is effective because it is, it's in the name, cytotoxic. Or in other words, it kills cells. Cyto means cell, toxic, killer. So and this is most effective against intracellular things. Over here makes sense, it's better against extracellular stuff that's floating around. But intracellular, let's say that here's a row of body cells, and let's suppose that we have a virus this time. Here comes our virus. It's going to come on in, and it's going to head into the cell. So this cell right here has now been infected by a virus. And the cell, because it's now been infected, it's going to start showing little pieces of the virus on receptors up here. It's going to show this antigen right here. So this T cell saw its target. Let's say this was a virus instead. So it sees a piece of the virus from the macrophage, and it comes over to the helper, and the T cell's like, I am looking for this piece of virus. So off it goes, it comes along, and so here's our T cell, and it comes and it's looking for that particular piece of antigen, that particular virus piece. And when it finds it, it hooks onto it and says, oh, here's my target. This is what the macrophage told me to kill. And then it hooks on, and because it's cytotoxic, it actually sends in poison into the cell, called perforins and granzymes, but basically it pokes a hole, and then it sends in a poison that is going to kill this cell. So the cell's now dead, virus is destroyed, we're happy and healthy, moving on. So there you have, in a nutshell, some basics on how B cells and T cells get activated, and then once they're activated, how the antibodies or the cytotoxicity work in order to actually protect us, and it's incredibly effective.